So why does DNA need to replicate? Well, when cells divide, each daughter cell needs an exact copy of the DNA in order to function properly. So during the S phase of the cell cycle, DNA replicates. In order to understand the DNA replication, you have to understand the structure. DNA has bases of adenine parethymine and guanine pyrazine, all connected by hydrogen bonds. It also has a sugar phosphate backbone, which we read 5' to 3', and the anti-parallel side is 3' to 5', all connected by covalent bonds. The first step is that an enzyme called Helicase comes in and unzips the DNA. It does that by breaking the hydrogen bonds between the bases. Then an RNA called primer comes in and places primase on the strand. The primase will tell the DNA polymerase where to start on the next step. Then a DNA polymerase comes and starts building where the primase was placed. It can only go the 3' to 5' direction while building the new strand 5' to 3'. While building, you have the leading and lagging strand. The lagging strand has Okazaki fragments because since the DNA polymerase can only go in one direction, it has to keep on going back as the helicase is unzipping the DNA, leaving gaps in the DNA backbone. To join the pieces together and fill in the gaps, an enzyme called Ligase comes in and fills in those gaps so that in the end, you have two identical DNA. The new DNA strands each contain one strand from the original DNA and a new strand created by a DNA polymerase. To recap DNA replication, the first step is helicase, which unzips the DNA by breaking the hydrogen bonds. And then an RNA called primer comes in and places primase on the DNA strand. Then the builder, DNA polymerase, comes in at the primase and starts building. And after all that, you have the leading and lagging strand, and ligase fills in the gaps on the lagging strand. And the result is you have two identical DNA